have a question.
Sure.
For the natural selection problem and the Hardy that problem, were we supposed to email the answers to you? Because I was a little confused.
Yeah, I think you'll just have to email them. I couldn't list them as a regular assignment because I would have had to do that way back when we first posted that. So just send it as an attachment.
So it's not too late to turn them in?
Nope, you can go ahead and turn them in.
Okay.
Any other questions? Something to keep in mind about assignments is when you turn them in, if you turn them in either a Microsoft Word doc or docx file, or if you turn them in Pages, which is a Mac file, I can make corrections directly on the assignments. So then if you want to see your assignments, you can always email me and I can just email you back the corrected version and you can see what you missed. Some people turn in PDFs. If you turn in a PDF, I can't really write on a PDF, so you won't be able to see the corrections. If you can send it in doc or docx or Pages, those are probably the best files. Or text files work too, TXT or RTF, which is a real text file. Any other questions?
Excuse me, sir, please.
Mm-hmm.
The assignment, is it for the gene pool Alehad frequency?
Right, so problems 5 and 6 on the Hardy-Weinberg problems. Those don't have pictures with them, so they just give you some numbers to start out with. And then problem C from the natural selection problems.
Question 5 and 6 and question 6. Okay.
So you're probably, when you answer those, just put them in a attachment and email them to me.
Okay, sir. Thank you.
You're welcome. Any other questions?
Yeah, was there a space on DTL to drop the assignment off, or?
No, because actually, since that was from some stuff that I put up about 2 weeks ago, and I didn't want to make it an assignment then because we were going to work a few first. So just put it in an attachment and email it to me.
Gotcha. Thanks.
Any other questions? Okay, I'm going to go to screen sharing and I'm going to look at the kind of sample questions that I sent you to view. So you should be looking at a picture of a walking stick right now, I think. So I'll probably wait till tomorrow when I get most of the, hopefully I'll get most of the Hardy-Weinberg and selection problems so I can give those back to you first. But then I'll post a big set of questions on evolution and taxonomy. And those will be due a week from tomorrow. So try to get those assignments into me today and then I'll try to get them back to you, or at least get them graded. If you want them back, just email me and I'll send them back to you corrected. As I mentioned, if you have them in Pages or Doc or DocX or RTF or TXT format. If it's PDF, then I can't make corrections on the document. So let's look at some of the sample questions. And I would ask you to, if you can, if you didn't have a chance, please look at the first 9 questions. These are all multiple choice questions. And we'll look at those for, let's take maybe about 5 minutes. My computer says 9:05, so we'll wait till 9:10. Let's take about 5 minutes and let's come up with the best answers you can, and then we'll go through them and we'll look at how this would be graded if it were on the test. Notice that the instructions for multiple choice questions are generally a little bit different. Notice the instructions say underline all correct answers. There may be more than one correct answer, or no answers may be correct. So you've all had questions where the last answer on the question is all of the above. On these questions, if it's all the above, there is no all the above. If on number one, if you like all the answers, choose A, B, C, and D, and E. If you don't like any of them, don't choose any of them. If you like A and D, choose A and D and don't choose the other ones. So notice the instructions say underline all correct answers. I sometimes have people highlight them. If you highlight them instead of underlining them, that's fine. If you want to see your corrected questions, it's usually best if you don't use red or blue ink on your answers, because that's what I use for the corrections. So you may have a hard time telling my corrections from what you did. So let's look at these. Let's take about 5 minutes and then we'll go through and we'll see if these 9 questions were some of the questions on the test. We'll see what your score would be, and then we'll look at the short answer question. And then we'll talk a little bit about the types of questions you might see on tests in general. So take a few minutes. If you didn't download this the other day, download this. It should have been in an attachment in an email that I sent you on Saturday or maybe Friday, I'm not sure. So download this, make some selections, choose your answers, and we'll see what the correct answers would be. So
Dr. Griffith,
Mm-hmm.
yes, are we supposed to answer the rest of the Hardy-Weinberg problems?
If you want to work on them for practice, you can, but you don't have to. If you want to work on them for practice and then you want to double check your answers with me, send me your answers and I'll tell you whether you're correct or not.
Okay. Okay, thank you.
You're welcome.
And all of them are in good areas, except for a few of them, two of them. The rest of them are in good areas. Only two of them are in bad areas. The rest of them are.
Jade, you may want to mute yourself.
But I mean, it doesn't make them. They didn't say nothing negative about her because she didn't want to go in the area.
Okay, let's look at the correct answers. What's an important part of Darwin's theory of evolution? Well, remember when we talked about his basic assumptions, differences between individuals was one of the things that was important. Natural selection was another one. So on this one, there are two correct answers, C and D. So for now, if you wrote down your own answers, just, put a check mark or do something to indicate if you didn't pick C or D, you would have missed that answer. If you picked A or B or E as a good answer, you would have missed those. Remember, A is really natural theology. And B is what Buffon said. And C is what Lamarck said. And so if we look at 2, why is polyploidy less common in animals than plants? Plants are rooted in one location. Plants are capable of photosynthesis. Animals aren't. Plants tend to be smaller than animals. I would say none of those is a good answer. Remember that the real reason why polyploidy is more common in plants is most plants have both male and female reproductive organs. So if they suffer a mutation where they get extra chromosomes, probably all the cells, the male cells and the female cells, suffer the same mutation, unlike in most animals. So that would be an example where no answers are correct. Gradualism proposes that evolutionary change involves long periods of time during which a species changes very little. Well, remember, that's the, should just say gradualism, not gradual equilibrium. That's a typo. Notice on tests, if you can find a typo or something like that, if you can find an error, you can underline the error or highlight it, and at the end of the question, point out what the error is, and you can get an extra credit point. So long periods of time during which a species changes very little, that's punctuated equilibrium. Rapid changes that occur in a short time, that's also punctuated equilibrium. Gradualism is small, slow changes in the gene pool over a long time. Keep in mind as we're going along, if you have any questions about any of the answers, go ahead and ask them. Which level of classification includes all the others? Well, notice that when I write these questions, I try not to give you a hint about whether there's one answer or more than one answer or 0 answers. But sometimes is a hint that's kind of built into the way the question is worded. So if you think about this, what level of classification includes all the others? There's really only one that could include all the others, unless we have 2 levels that are at the same point. But otherwise, there's only one. And in this case, the one would be a domain. Now notice if domain wasn't listed, suppose the answers we had were just A and C and D and E and F and G, then the answer that you would choose would be kingdom. Because notice it doesn't say which level of classification is the highest level. It just says which level includes all the others. So that means if you look at all these, what could you put all these other levels under? So if domain wasn't listed, it would be kingdom. If we just had 2 levels, if genus and species were listed, then genus would be the level that includes the level of species. K5, a Hardy-Weinberg problem. If 36 out of 100 individuals in a population show the recessive phenotype, what's the frequency of the recessive allele? Well, remember, 36 out of 100 isn't the frequency of the recessive allele. It's the frequency of the recessive allele squared. So you have to take the square root of that to find the answer. So that would be 0.6. And again, although I try not to give you any clues about how many answers there are, if you think about most questions that are mathematical, there's probably only one answer. It's hard for the answer to be 2 and 6 or whatever. It's not very feasible. Which level of classification has a clear biological meaning? So remember what we said? Domains are based on the types of cells that we see. So they have a clear biological meaning. We said all the other intermediate levels have more of a subjective biological meaning. An order might mean something different to you than it means to me. What does natural selection act on in a population? Well, if you think about it, we focused mostly on gene pools, how allele frequencies are changing. But if you think about our peppered moss and you think about the pictures we looked at, natural selection is what's eliminating some individuals and allowing other individuals to survive. Well, if we think about peppered moss, moss survive because of their coloration. That's phenotype, that's not genotype. So natural selection actually eliminates some individuals or allows individuals to survive based on their phenotype. Ultimately, we're interested in the genotype changing, but it's really the phenotype that's impacted by natural selection directly. If 49 out of 100 individuals in a population show the recessive phenotype, what frequency is heterozygotes? Okay, so this one we have to think about this. 49 out of 100 is the recessive allele. So we know that Q squared equals 0.49. So Q squared equals 0.49. Remember, Q is the recessive allele. So if we take the square root of both sides, Q is equal to 0.27. But this isn't asking for Q. This is asking for the frequency of the heterozygotes. So we have to find 2 times P times Q, or 2 times, if Q is 0.7, then P is 1 minus Q. So P is 0.3. So 2 times 0.3 times 0.7. And if we multiply that out, I think we're going to get 0.42. Which geological era is referred to as the age of reptiles? So you probably will have to look that up on the geological time scale that we had, or another one. And this is the Mesozoic. Okay, so let's look at all these problems and see how this would be graded on the test. So first, let's look at the total number of answers. Okay, there's 5 answers in number 1, 3 in number 2, so that's 8. 3 more, that's 11. There's 7 in number 4, so that's 18. There's 4 in number 5, that's 22. There's 6 here, that's 28. There's 2 more in 7, that's 30. There's 4 in 38, that's 34. There's 4 more in number 9. So if I added right, I think that's 38 points. So these multiple choice questions would be worth 38 points. you would get 1 point for every correct answer you picked. So every one that we indicated in blue. You would get 1 point for every incorrect answer you didn't pick, all the ones that are still in black. If you picked an answer you shouldn't have, like if you picked A here on number 1, you would lose a point. Or if you didn't pick a correct answer, if you didn't pick C. So your score on this would be 38 minus however many you had missed. So over the years, I've had students tell me that the best way to answer these questions is, if you're not sure, don't choose anything. If you're sure, choose it. If you're not sure, choose everything. Of course, I'm going to tell you the best answer is to be sure when you choose your answers. Probably overall, if you look at the questions, there's more questions that just have one answer than anything else. But there typically are some questions that either have no answers, like number 2, or have multiple answers, like number 1. The best advice I can give you is to think of each question, if you kind of put it with the answer, think of it almost like a true-false question. So number 1, what's an important part of Darwin's theory of evolution? So look at A, creation of living things by God. Was that an important part of his ideas? If it was, choose that answer. If it wasn't, don't. And if you choose A or if you don't choose A, it has nothing to do with whether you choose B or you don't choose B. So B is a completely separate proposition. So don't think about what you chose for A when you choose your answer for B. So this will give you a feel for kind of how you do this. Notice on the test questions that I'll send you that you'll have a week to work on, if there's a Hardy-Weinberg problem, and the problem will tell you this, but it will say, show your work. So, write out the formula that you're using, substitute the numbers in, and then give me your numerical answer. If you just write down your numerical answer, you'll probably lose 2 points, for not having the general formula and not substituting the numbers in. So make sure that you do that. Now let's look at short answer questions. And keep in mind on the questions that I give you that short answer questions usually are short answers. They don't take more than a couple of sentences. If you find yourself writing a whole page, unless you really write large, you're probably giving way too much information. Make sure that when you answer questions, you put them in your own words. If you do copy something directly from Wikipedia or someplace like that, and I detect it, you get a 0 on the whole assignment. So you have to word things in your own language. If you're not sure how to do that, there is a writing center, and they do have online appointments because of the environment that we're in. Check with them, show them the source where you're getting the information for your answer, and ask for some help in how you word that. So let's look at how you could answer a question like this. Describe how the evolution of camouflage in a prey organism like a walking stick insect illustrates Darwin's observations and conclusions about nature. So up on the top of this picture, there's a branch, and down below, hanging onto this branch, is an insect. You might be able to see the long legs that are attached. And I think you can see pretty clearly why this is called a walking stick insect. Like a lot of organisms that aren't poisonous, walking sticks are camouflage. They try to blend in with their environment, so nothing that eats them will notice them, kind of like those peppered moths that we talked about a few days ago. If you're poisonous, you want everybody to notice you so they don't eat you. If you're not poisonous, you don't want them to ever notice you. So Darwin's observations, Now, one of the things you have to keep in mind, a mistake that I saw students make a lot in Biology 105, and I am stricter on how I grade the essays in 106 than I am in 105, but people give vague answers. Remember, whenever you're thinking about science, you have to think about details. So think about Darwin's observations. One of his observations is there's too many offspring born for all of them to survive. So what you're going to do is you're going to take Darwin's observations and you're going to apply them to walking sticks. And as you can see with my typing ability, it is good to go back and double check your answer when you get done. Not only might you make a typo, but even if you don't make a typo, sometimes autocorrection will change what you wrote. So there are too many walking sticks for all of them to survive. So this is one of Darwin's points. Now, remember then, Darwin said when we look at all the individuals in a population, they're all different. So in this case, we're focusing on these, on their camouflage. So you could say something. These walking sticks are different in how good their camouflage hides them from predators. And keep in mind, there's almost always more than one way to say this. You could say that some walking sticks have good camouflage and are hard for predators to see. Some walking sticks have poor camouflage and are easy for predators to see. Now, remember what Darwin said. Some of these differences, in this case, we're talking about camouflage, are important. So camouflage is important. For the survival of walking sticks. Darwin's other observations, well, remember, he said things that are going to evolve have to have some kind of genetic component. So you could say that camouflage is partly inherited in walking sticks. You could just say it was inherited. I'm not sure it's completely inherited, so that's why I put partly. And then remember, he had one more observation, which is there aren't enough resources to go along. So you could say there, and one of the things I would recommend is that you kind of look at your answer when you get done before you turn it in. Like I would say that sentence really fits better up here when you say there are too many walking sticks for all of them to survive. Why? Well, really because there aren't enough small twigs and branches for them to hide on. If there were plenty of twigs and branches, maybe the population size could be bigger. Now, what's Darwin's conclusion? Well, remember, his big conclusion was descent with modification. The next generation is different than the previous one. So you could say something like, next generation of walking sticks be better camouflaged than this generation. But notice Darwin doesn't say that they're all going to be the same. I guess I should say there will still be variation. So you would have to do something like this. Now, if this were on the test, if this were the question, this would probably be maybe 8 or 9 points. And so one of the things this means is if you look at short answer questions on the test, you can get an idea of how many different points you have to make. And the way you can do this is there's usually 2 or 3 points. Spelling, grammar, punctuation, and things like that. So if you take 2 or 3 points away, notice that leaves 6 points. So one of the things that that should tell you is you're going to have to tell me about 6 things. So there are too many walking sticks for all of them to survive. There aren't enough small twigs and branches for them to hide on. The walking sticks have different camouflage abilities. Okay, that's 3. Camouflage is important for survival. That's 4. Camouflage is inherited. That's 5. The next generation has better camouflage. That's 6. So if you see something like this and it says 8 or 9 points and you say one thing, you say there's too many walking sticks for all of them to survive and you stop, you're probably going to miss, a lot of questions or a lot of points on that question. Now, besides these types of questions, there's a short answer question on almost every set of questions that we have this semester, sometimes 2 or 3. Besides that, usually most of the points on the test are multiple choice questions. There are typically a few true-false questions. I don't like true-false questions a lot, but sometimes there's not another way I can think of to word something that I want to ask. Keep in mind, I always tell students, there are some studies that show that on a true-false question, if students don't know the answer, they tend to pick true. Why do they tend to pick true? Because they don't want to believe that their nice, generous, kind biology teacher would tell them a lie and put a false question on the test. But if your nice, kind, generous biology teacher knew that people almost always pick true if they're guessing, they might put some false questions on the test. Or they could just tell you that to mess with you too. Keep in mind, it could always be better. In one biology class I took, I had a final exam that was 200 questions and 192 of them, it was all true false. Out of the 200 true false questions, 192 were false. And after you did about 7 or 8 questions in a row that you knew were false, as soon as you got to one that you got a question about, you automatically picked true because you knew it had to be true. You hadn't had a true answer in a while. And you didn't have to have a true answer apparently, because there was only one about every 25 questions. Really a horrible test. Besides a few true false, you might have some matching. Make sure if there is matching that you read the instructions carefully, because sometimes in the matching questions, you can only use the answers once. Sometimes you can use the answers more than once. So that might make a difference in terms of, how you want to go about analyzing those questions. There are sometimes sequencing questions. We'll talk about some processes that have multiple steps, and you might have to put the steps in the order that they occur. Or you might have to know which step is between 2 other steps, something like that. Now, the other thing that you can potentially have on questions is you might have a picture and have to identify something about that picture. In the next section where we start looking at biodiversity, the different types of living organisms, you'll have some questions with that on them. It'll be a picture of something, and you'll have to tell me what type of bacteria cell it is, or something to that effect. Besides that, you could have some diagrams rather than photos that you have to label things of. People who were in my 105 class, we had, if you remember, we had some drawings of mitochondria and chloroplasts. And you had to either name different parts of the drawing, or you had to tell what happened there. Was it the light-dependent reactions, or was it the light-independent reactions? Was that where glycolysis happened, or was it where the Krebs cycle happened? So you could have a diagram. And the other main type of problem that we have is something that you see here kind of in a multiple choice format. You could have some problems. I guess I would call them math problems, basically. I can guarantee you on this test, you'll have a Hardy-Weinberg problem. And then that problem will have several sub-problems, A, B, C, D, E, F, where you'll be given some information at the beginning, and then you'll be asked, well, what do you predict for the frequency of heterozygotes? What do you predict for how many individuals have the dominant allele? Those sorts of questions. And then on this test, you'll have a second math problem. You'll have one with natural selection, where you'll have some raw numbers before selection and selected. And then you'll have some, have to calculate the numbers after selection and calculate the frequencies and answer questions similar to the ones on those natural selection problems. We worked one in class, and then I asked you to work two. So as I said, I'm hoping you all get that to me today, and then I can send those back to you tonight or tomorrow morning, and you can kind of use those as a guideline to study for the test. So now, even though I just called it a test, it's really just questions. Unlike some online classes, this is not timed. So when I post these, you'll have a week to work on it. You can work on a little bit here, a little bit there. But do keep in mind when you turn in the test, if you turn in the test and late, it's 10% off. And then if you do it a second time, it's 20% off. So you definitely want to turn it in at the correct time. And obviously, since everything's on the computer, everything you send in is time-stamped. So, you know, keep that in mind. Are there any questions about what you might see on the test? I guess the one other thing I should mention is the test may have two parts to it, or it may have one part. Normally, my big question sets are close to 200 points. I'm not sure if D2L will let me do that. If it'll only let me give you 100 points on one assignment, then the questions will probably come in part one and part two, with, you know, half the questions on one set and half the questions on the other one, if I can't go over 100 points on a single assignment. So again, people who had me for 105 might remember at the end of the semester, I think we had a set of questions on DNA and a set of questions on genetics that were both divided into part one and part two. So it'll be similar to those. Any questions about the exam? Okay, I'm gonna stop sharing this screen if there are no questions. Don't forget to give me the Hardy-Weinberg problems and the selection problems sometime today, please. If you don't give them to me till late, keep in mind you probably won't get them back for a little while to look at them. I'm going to start a little bit on the next section we're going to study, and hopefully some of this will be review. You should be looking now at a bacteria cell. And really for about the middle 6 weeks of the class, we're going to be focusing on biodiversity, the different groups of living organisms. So we're going to start with the domain bacteria. And remember that the main characteristic of the domain bacteria is that organisms in this domain have prokaryotic cells. So what's a prokaryotic cell? Well, cells that don't have a nucleus, cells that don't have membrane-bound organelles. So I'm sure you could all name a bunch of membrane-bound organelles, I hope. Mitochondria, chloroplasts. The nucleus is actually a membrane-bound organelle. Smooth and rough endoplasmic reticulum, the Golgi apparatus, vesicles, peroxisomes, lysosomes, the central vacuole in plants, other types of vacuoles. So that's a few types of membrane-bound organelles. There are some others. So let's look at a bacteria cell and remind ourselves. We talked about this back when we talked about prokaryotic cells in 105 in the chapter on cells. So if we look at our bacteria cell, first, let's focus on the things that we find in every bacteria cell. And there are really only 4 of these structures that we find in every bacteria cell. Towards the center of the cell, we have a region called the nucleoid. Now remember, oid means like, so the nucleoid is like a nucleus. And it's mostly like a nucleus because this is where the genetic material is. Now, in a prokaryotic cell, there's only 1 chromosome, as opposed to eukaryotic cells like human cells that have 46. And almost all eukaryotic cells have an even number. And that's because the eukaryotic cells have the capability to reproduce sexually. Prokaryotic cells only have 1 chromosome. They cannot reproduce sexually. They can reproduce asexually, which means that the daughter cells that they make are genetically identical, unless there was some problem with replication of the DNA. They can go through sexual processes. Sexual processes in bacteria change the DNA of a cell. So they alter the DNA, but they don't produce a new cell. Reproduction is producing a new cell. So notice sexual processes and reproduction can be separate, like in bacteria, or they can occur simultaneously, like in people, where when we have meiosis, crossing over, fertilization of an egg by a sperm, not only do we get reproduction, creation of a new cell, but we get genetic variation. That new cell does not have the same DNA as the parent cells, the egg and sperm that produced it. So in bacteria, sex, reproduction, completely separate. They don't occur together. So we want to keep that in mind. So we have 1 chromosome. Now remember, not only is this chromosome different from eukaryotic cells in the sense that there's only 1, but remember that this is a circular molecule. Now the DNA in this chromosome is made up of smaller units called nucleotides. Remember, the nucleotide is the deoxyribonucleic sugar, the phosphate group, and then a nitrogenous base, either adenine, cytosine, guanine, or thymine. And in a circular molecule, the last nucleotide is bonded back to the first nucleotide. Now, as you can see from this purple glob in here, circular is a little bit of a misnomer. It's kind of twisted. If you ever saw those cartoons with Charlie Brown's kite string, they kind of look like that. So this is different. In eukaryotic cells, the chromosomes are linear. So circular chromosome in a prokaryotic cell, linear in a eukaryotic cell. And this is a molecule that's made of naked DNA. And what we mean by that is it's DNA, and the DNA is all by itself. Remember that in eukaryotic cells, the DNA is wrapped around histone protein, kind of like thread around a spool. So the chromosome in a bacteria cell is different from the chromosome in a eukaryotic cell in several different ways. Now, despite the fact that this chromosome is different in several different ways, it's similar in one very important way. This chromosome can be divided up into little sections of DNA, and the sections of DNA have the instructions for making proteins. And these sections are called genes. And then remember when we talked about operons at the end of 105, there are also some things called operators and promoters. Operators are like an off switch, and they stop the cell from making messenger RNA and ultimately protein from genes. Promoters are on switches, and they turn on those genes so that the cell uses them to transcribe messenger RNA and then to translate protein. So although the DNA is different in a lot of ways structurally, functionally, it works the same as the DNA in a eukaryotic cell. Now, that's one of the things that we see in a prokaryotic cell. Out around the nucleoid, we have this kind of light yellowish, orangish material in here, and this is the cytoplasm. Now, if you were to look at the cytoplasm, the most common molecule in the cytoplasm is water. But besides water, there are a lot of chemicals. There are enzymes that are floating in the water, enzymes for the various metabolic reactions that this bacteria cell is going to carry out. There are reactants for those metabolic reactions. There are the products of some metabolic reactions that have just completed. There are ATP molecules to provide the energy for metabolic reactions. There's ADP and phosphate groups left over from that ATP that's already been used up to provide energy. There's glucose so that the cell can carry out glycolysis and, for a lot of bacteria, aerobic cell respiration to make more ATP. There's all sorts of different molecules in this water. So although water is the most common molecule, it's not runny water. It's sometimes described as having the consistency of a milkshake or of a kind of a thick jelly. So it's kind of semi-liquid, semi-solid. So even though water is the most common molecule, you don't want to think about this as being runny, as being liquidy, because it's not. I think we're about out of time, so we're going to stop here for today. Are there any questions about bacteria cells before I go away from this screen? We'll get to some of the parts that I haven't talked about yet Wednesday. If there aren't any questions, we'll leave this. And if you have any questions about anything else, you can ask them now. Otherwise, I'll let you go. I'll post this video later. And if you check your grades for the monohybrid cross should be up, except there's about 2 or 3 people that it looks like it didn't take. I think I forgot to save it after them. So I'll get those up in the next couple minutes. You can look at those. As I said, if you sent me a Word document or a Pages document and you want to see any corrections, I can send those back to you. If you sent me a PDF, that's the big advantage of sending me Word or Pages, is I can make corrections and then send those back to you. I can't do that on a PDF. If you don't have any questions, you're free to go. And don't forget, we don't have class tomorrow, so I will see you Wednesday.
So Griffin,
Mhm.
the question number 5 and 6 you asked us to do.
So
Yeah.
5 and 6 on the Hardy-Weinberg problems and C on the natural selection problems.
Yes, are we supposed to turn it in before the class? Like, are we supposed to send them to you?
You can just send it to me as an attachment. I'll still accept them, so you're fine. Today would be good though, because I'm probably going to post the big question set tomorrow. And if you send them to me today, I'll try to get them back to you so that you can use them as a guide to answer similar questions when you get the big question set.
You send it to your email, right?
Yeah, just send it to my email, it's fine.
Okay, thank you.
You're welcome.
Excuse me, sir.
Mm-hmm.
Yeah, the frequency selected. I didn't understand in the video how you worked it.
So for any of the frequencies, the first thing you have to do is add everything in that row going across together. So you're going to add all the numbers that were selected together. And so you're going to get your sum, and maybe your sum is 300, I don't know. So then the frequency of number one is going to be how many number ones do you have divided by 300 divided by the total that were selected. And then your frequency of number two will be how many number twos do you have divided by the total that were selected.
Okay, is it the total of the number 2 divided by what? Divided by 2.
So for the frequency of number two, there's going to be a number in the number two box.
Yes.
So take that number and then divide it by, add up the ones and the twos and the threes and the fours and the fives and the six all the way across. So divide it by whatever that total is when you add everything up.
Okay. Thank you.
And again, if you want to double check when you get done, after you figure out the frequency of number one and number two and number three and all of them, it should add up to about 1. Now, depending on where you rounded, it might add up to like 1.01 or 0.998 or something, but it should be pretty close to 1.
Okay. Thank you.
You're welcome.
Professor,
Mhm.
how many points was the monohybrid cross assignment? I'm seeing 11 points in one section and 12 points in another section.
Should be 12 because there were 12 questions. So I'll check and see. I think I may have said 11 first. I don't know how I couldn't count that there were 12 questions, but it's 12 in the grade book, I believe.
Okie doke. Thanks.
You're welcome.
Do you have any problems with email going coordinating with DTL at all? I've been
I've had some occasionally. I think most of the time, if you submit something to me in D2L, it comes to me at my regular school email. I haven't noticed anything that didn't. But if you send me something through D2L and never hear from me, then let me know, because it might be that it didn't come to my school email, so I never saw it.
It's happened to me a couple times already. Like I sent someone something through DTL and it doesn't show up, and I have to separately do it through the school email.
Okay. Yeah, you can always try that if it doesn't work. I would say try D2L first, just because I don't know how
And then
your
I get
email
things
is, but
that like aren't that I don't see on DTL, but then I see in my school email.
oh, that's weird.
Yeah, and which isn't great because I don't check the school email as often as the DTL for sure.
Well, and that's why I try to like always post our invitations and most of the things on the class page, and then also send you an email, but that probably is going to your school email. I don't know, but I'm guessing. You might want to at least check it, once every couple of days though, because I do know that, if there's no classes allowed on campus or something, if there was a positive COVID test for somebody who was on campus, that's usually where that information is posted.
Okay, doke. Thanks.
You're welcome.